Familial Hypercholesterolemia by Hannah Cole, Kristen Vinson, and Sierra Smith. I'm going to start by giving you a little bit of background on familial hypercholesterolemia, commonly referred to simply as FH. This is an autosomal dominant inherited trait that can occur in either the heterozygous or homozygous form. Upon its discovery, this genetic disease was originally thought to have been controlled by a single gene, the low-density lipoprotein receptor gene. However, more recently, new genes such as alpha-lipoprotein B100 have been found to be a cause of this disease as well. Currently, heterozygous familial hypercholesterolemia is one of the most common inherited genetic diseases. However, getting a proper diagnosis can actually be very difficult. This is largely due to the fact that many genes that cause the disease have not been discovered yet. Now, what does FH do? Well, the most common physiological effect is very high levels of low-density lipoprotein, which ultimately leads to cardiovascular disease and atherosclerosis. Additionally, xanthomosis are commonly seen, particularly at the Achilles tendon. These are actually hard nodules of cholesterol buildup that can be seen with the naked eye. So what was the process of figuring out this disease? Well, as early as 1873, clinicians such as Faggy had reported single cases of xanthomosis and cardiovascular disease. However, it wasn't until much later in 1937 when Norwegian clinician Carl Molar put it all together. He had the idea that hypercholesteremia and tendinous xanthomosis were linked to cardiovascular disease through a single inherited gene. He came up with this idea because he noticed patterns in 17 families over 4 generations. In 1964, 2 forms of FH were described, the heterozygous and the homozygous forms. Now, prior to this, it was only known as FH. There was no knowledge of a mild form versus a severe form. 1973. This is when the mechanisms of the disease are solidified. Assistant professors Michael Brown and Joseph Goldstein conducted research, and they were able to show that defects of the gene that codes LDL receptor protein caused FH. Their work won them the Nobel Prize in Medicine in 1985. Research of this common genetic disease continues to this day. Scientists are continuing to discover previously unknown defective genes that cause FH, and there has also been much research into effective treatments for this disease.
Now that we know the history of FH, we can define it as an autosomal dominant genetic disorder where there's a mutation in specifically one or more of the 3 genes listed here. The mutation results in the individual being unable to regulate and remove cholesterol from their plasma, which is the liquid part of the blood. The cholesterol buildup in the plasma is attributed to a high concentration of low-density lipoprotein cholesterol, and we're going to look more into the genes as well as these lipoproteins in the future slides. We're first going to look at lipoproteins, which transport cholesterol in the plasma. So every individual will possess both high and low-density lipoproteins, and I will first define high-density lipoproteins. And they're considered to be good in excess because they carry cholesterol to the liver where it can be digested and then excreted from the body. This results in the plasma being low in cholesterol, which then reduces the risk of plaque buildup in the arteries. The second type of lipoprotein is called the low-density lipoprotein, and these are also considered to be good because they take cholesterol to cells where it can be used for membrane fluidity or even synthesizing hormones. The low-density lipoprotein cholesterol moves to a cell by being pumped through the arteries. However, the drawback to this is that in high concentrations of low-density lipoprotein cholesterol, the cholesterol is not being digested and excreted from the body. It is rather being circulated and deposited in tissues. So if we look in Table 1, an individual that possesses FH will have much higher levels of low-density lipoproteins and much lower levels of high-density lipoproteins compared to a normal individual. We are now going to look at different gene mutation processes that result in FH. However, I first want to walk us through a normal process of a low-density lipoprotein cholesterol interacting with the cell. So if we look at the leftmost figure where it says normal at the very top, we have a blue LDL-C, which is our low-density lipoprotein cholesterol, and on the cell membrane, there is a black Y, which is our low-density lipoprotein receptor. So once the LDL-C finds the cell that sent a signal for cholesterol, it will bind to the LDL receptor. Then the LDL cholesterol receptor complex is going to permeate into the cell, which will result in the 2 detaching. The cholesterol will carry out its function required by the cell while the receptor is going to regenerate back to the cell membrane so that another LDL cholesterol can use it and this process can repeat. Now that we have an understanding of how LDL cholesterol is supposed to interact with the cell, we can look at different gene mutations associated with FH. The first gene mutation that we will look at is the low-density lipoprotein receptor gene. This process can be seen in the figure that has LDLR at the very top. A mutation in this gene results in a single amino acid change in the protein, which prevents the LDLC from binding to the receptor. Scientists have found over 2,000 mutations in this gene that result in FH, so it has an enormous diversity. The second gene mutation is the apolipoprotein B gene, and this mutation process can be seen where the APOB is written at the top of the figure. This gene is responsible for making two types of APOB proteins, APOB B100 and APOB B48. Both of these proteins are building blocks for lipoproteins. However, the APOB B100 protein is a building block for the low-density lipoproteins. If the APOB B100 protein is mutated, it will not be able to bind to the LDL receptor. Additionally, scientists have found over 100 different mutations in this gene that cause FH. Lastly, the third gene mutation is called the pro-protein convertase subtilskin kexin type 9 gene. This gene mutation process can be seen in the figure with PCSK9+ at the top. The gene mutation results in enhanced activity of the receptor, which speeds up degeneration, thus preventing the receptors from regenerating back to the cell membrane for future use. Additionally, scientists have found over 50 different mutations in this gene that cause FH. Overall, these gene mutations result in cholesterol re-entering the plasma. From there, the cholesterol will deposit into tissues such as the arteries or skin. And in future slides, we're going to look at the different phenotypic effects that this has on the body.
Familial hypercholesterolemia follows an autosomal dominant inheritance pattern. The mutated gene is dominant, meaning that if you are heterozygous for this disease, you have a 50% chance of passing it on to your offspring. If you're homozygous for this disease, you have a 100% chance of passing it on to your offspring. In the pedigree I have in the bottom right corner, this is an example of a family that does have familial hypercholesterolemia, and you can see that it's equally divided among males and females, and it does appear in every generation. Being heterozygous for this disease is more common and more manageable. Being homozygous is rare, but it can happen, and it requires more serious and immediate treatment.
Due to the different types of gene mutations in FH, cholesterol will not degrade and be digested. Rather, it will circulate in the plasma until it deposits in a tissue. If cholesterol deposits in the arteries, it's known as atherosclerosis, and it's seen in the bottom right corner of the slide. This can lead to chest pain or discomfort or even difficulty breathing due to less oxygen and blood flow in the body. Additionally, cholesterol can deposit into other tissues, and that is seen in figures A, B, C, and D. If cholesterol deposits under the skin, it will be called a xanthoma, whereas if a white cloud ring of cholesterol forms around the edge of the cornea, it is called a corneal arcus. Now, the xanthomas and corneal arcuses have no negative effects on the body. It's purely just cosmetics. However, it is a very good indication that the individual possesses high levels of cholesterol in their plasma.
Some ways people can get diagnosed with FH is through a physical examination. The cholesterol will build up and you can physically see the cholesterol deposits on the skin. You can also do lab testing. Genetic testing is available. There's an FH panel that will test the LDLR, the APOB, or the PCSK9 gene to see if a mutation is present. You can also do blood testing to see the LDL levels in your blood. This picture shows a comparison between people who don't have FH to people who do, and you can see that the LDL cholesterol levels, the average is much higher in people that have FH. And early diagnosis and treatment is really important for people that do have familial hypercholesterolemia. FH is not a curable disease. There are, however, effective treatments available, one of them being statins. Statins are a prescribed medication that help decrease the amount of LDL cholesterol in the body. And they're able to do this by blocking enzymes that are needed for cholesterol production. They're very common. I have a very common example that is oftenly prescribed in the top left corner of this slide. It's known as atorvastatin. Statins are effective, but working alone for people that have been diagnosed with FH, they're not strong enough to bring down the LDL cholesterol levels to a normal range. In serious cases, they have been known to cause liver damage and adverse neurological effects. Another example is bile acid sequestrants, which either come in pill or powder form. They're often used in combination with other treatments, and they work by lowering the amount of bile acid that goes to the liver so that the liver has to use cholesterol from the body in order to convert to bile acid, which in return lowers the cholesterol levels. On top of everything, diet and exercise are very, very important for people that have been diagnosed with FH. It's important to maintain a healthy lifestyle and try and eliminate any outside source of cholesterol that you could be getting. I have some examples down here of heart-healthy foods that have been known to lower cholesterol levels, and also some exercise tips of some exercises that have been known to lower cholesterol levels. Some more treatment options available are injections, and an example of an injection is a PCSK9 inhibitor. These inhibitors work by stopping PCSK9 proteins from functioning, and the PCSK9 proteins often interfere with receptors, and they stop the receptors from being able to sweep the extra cholesterol. So when the protein's not functioning, it allows the receptors to do their job better. It's reported to decrease LDL-C levels by about 60% after treatment, and it is mainly prescribed to people who can't take statins because of the side effects that statins can have. There's also an ongoing clinical trial from the Mayo Clinic that is using a biobank to help further target individuals that can use PCSK9 inhibitors, as well as researching to make the inhibitors themselves more effective. Evkeeza is an injection that was just FDA approved this year. There's still a lot of ongoing research with Evkeeza because it's the first injection that actually binds and inhibits proteins that aid in lipid metabolism. It's mainly targeted for individuals who are homozygous for FH, and it decreases the LDL-C levels by about 49% after treatment. A more serious treatment option is LDL apheresis, which works very similarly to kidney dialysis. It helps filter out the LDL cholesterol from your blood and then returns your blood back to you. It's often very effective, reducing the LDL-C levels by about 70% after treatment. And low blood pressure is a serious side effect from LDL apheresis. In serious cases, a liver transplant might be the only option left that can help manage the disease.
We've covered a lot of information about this genetic disease. Now I want to give you some additional tidbits. Like many other diseases, this one is referred to by more than just one name. As I'm sure you've noticed in this presentation, both familial hypercholesteremia and FH are used interchangeably. Some other names this disease is commonly referred to include autosomal dominant hypercholesteremia, hyperlipoproteinemia type 2A, as well as other names that reference the specifically affected gene, such as LDL, ApoB, or PCSK9. However, no matter the name being used, this is an autosomal dominant disease that affects the amount of low-density lipoprotein cholesterol buildup in the body. The wide variety of names may be due to the fact that FH is one of the most commonly inherited genetic diseases. Although the numbers vary worldwide, it breaks down to roughly 1 in 200 people have a mutation of a gene that codes for the clearance of low-density lipoprotein cholesterol from the bloodstream. Now, to put that number in perspective, as of 2020, there were 1.3 million affected people in the U.S. alone. Now, anyone may be affected by this disease so long as they have a mutated gene. However, research has shown that the occurrence of this disease may be more common in certain groups of people, including the Ashkenazi Jews, some Lebanese groups, and French Canadians. From this data, it appears that this genetic disease has no particular origin. Although there is an equal probability for both men and women to inherit FH, men tend to be affected sooner in their lives than women. On average, men with FH will suffer a heart attack in their 40s or 50s, while women with FH suffer a heart attack about 10 years later, so in their 50s to 60s. Now, without a proper diagnosis and adequate treatment, both men and women suffer an MI sooner than this. However, the 10-year age gap between the two still remains the same.